Hello and welcome to this video notes of mitosis. What is it and how does it preserve the number of chromosomes in a nucleus? Okay, so we should know that mitosis is the dividing of the nucleus that preserves the number of chromosomes in the daughter cells. Whoops, we have a little typo here. We should have to preserve the number of chromosomes in the daughter cells. There we go. Okay, to understand our chromosomal number, we should first discuss what a zygote is. Well, the zygote is the fertilized egg. It is basically the first cell that has all of your chromosomes in it that's going to make you who you are. How is a zygote formed? Well, zygotes are formed from the two sex cells, sperm and egg. So here we'll have sperm from the male and egg from the female. And these two cells are both called haploid cells because they are what I call it 1N. So that's a 1N. This is a 1N. And 1N means one set of chromosomes. Well, what's our haploid number or 1N number for human sex cells? It is 23. So the sperm is going to carry 23 chromosomes and the egg is going to have 23 chromosomes. Well, what happens after fertilization? Well, during fertilization, the nucleus of a sperm cell combines with the nucleus of an egg cell. So we have one set of chromosomes from the male, which is now entered into the egg cell, and you have an egg cell that had the 23 chromosomes. So simple math is going to give us 23 plus 23. And the sperm is going to be 1N, and the male is going to, and the female egg is going to be 1N. So we're going to have a situation of 2N. So we have now we have a zygote, which is the full set of chromosomes that make a human what it is, and we consider this to be 2N. Two sets of chromosomes, one from mom and one from dad. And basically we have, this is called a diploid cell. Diploid. Diploid, di meaning two, and ploid referring to the number of sets of chromosomes. Well, now what? We have our zygote with our 46 chromosomes, two sets of chromosomes, two sets of 23, meaning it has 46 total. Well, what's it going to do? Well, we can't be just one cell. It has to divide. So after fertilization, this fertilized egg or zygote will do a number of cell divisions, and those are mitotic cell divisions, and this will divide into two cells that are both diploid. And we'll just put them down here. This will be a little smaller, but they will have 46 chromosomes in each of their nuclei. And this is mitosis right here. So this right here, mitotic cell division or mitosis. And virtually all of our cells, body cells or somatic cells, have come from this type of cell division. So these result in somatic cells, which virtually are body cells. They could eventually differentiate into everything from our heart and our lungs to our muscles, our bones, everything, every structure of really makes up our body. And don't forget, these somatic body cells are 2N. They are diploid. All of our cells, except for sex cells, are diploid. Well, now we have to look at how does the cells do that? How do they go from being diploid to one cell to being diploid to having two cells that are still diploid? Well, the answer is that the DNA replicates itself. And we know that replicating means to make a copy. So before the cell divides, it goes through this DNA replication during interphase, and then mitosis will then be at the after the interphase, and mitosis is a way of organizing the chromosomes and dividing the chromosomes up so that each cell has an exact copy. So in order to demonstrate how this happens in interphase and mitosis, and then eventually cytokinesis, and we'll wind up with two cells that are genetically identical and that are still diploid. We're going to start with this cell that only has two chromosomes because it's nice and easy to draw. Okay, well, like we said, during interphase, the cell is going to replicate its chromosomes. So what it'll do is it'll make a copy, and we'll go through DNA replication a little bit more advanced later when we get to genetics. But for now, it's going to make a copy, and it's going to keep the two copies together. And it attaches them with a centromere, and that centromere holds the two copies together. And we'll talk about that in a second. So this one will basically go in there, and again, held together by that centromere. Okay, so basically now we have two chromosomes still, but they each have an identical copy, and the cell is very smart in that it keeps the copies together. That's essential for making sure that each cell at the end of mitosis or end of cytokinesis has the genetic identical to the parent cell. Okay, so it's hard to draw this out in the DNA replication, but you have to remember DNA is double-stranded. It's going to unzip. It's going to then replicate. You're going to wind up with two chromosomes now that are genetically identical after DNA replication. I'm just going to draw in that second chromosome for now, and so I can label this, and make sure you label these in your notes as well. So know that these two chromosomes, okay, here and here, are called sister chromatids. Sister chromatids. And what they are held together by is a protein called a centromere. Centromere. Okay, I messed that. Okay, I think I finally got it. You can see that it's held together by a centromere. And that's exactly what we see in our nucleus over here. Okay. Well, we actually kind of skipped the step of the chromosomes shortening and getting more visible. So we actually are starting with prophase right here. So this is prophase. And during prophase, the chromosomes become visible. Yes, they look like those X's, but we talked about what they actually are. And then another thing will happen is that the nucleus will start to dissolve. So you can see the nuclear membrane is breaking down. Okay, and now what will be able to happen is the chromosomes will be able to migrate towards the middle of the cell. And I left out in prophase the centromeres, and these centromeres are on the opposite sides of the cell. So right here and right here are centromeres, and they were at closer to the nucleus when the cell wasn't dividing. And also we have, in addition to the centromeres, we have these other microtubules called spindle fibers. And the spindle fibers and the centromeres certainly aid in separating the chromosomes and getting them to migrate to both sides of the cell and getting to right location. Now, before we separate these chromosomes, keep in mind, remember, um, yes, they look like X's, but really what they are two sister chromatids attached to each other, and they are replicas of each other. When were they made? During interphase, when the cell was replicating its DNA, or DNA replication. So now what? Now the chromosomes are going to separate. The two sister chromatids will separate during anaphase. This right now is metaphase. This is the middle of the cell, but when they separate, they're going to be during anaphase. So here's probably middle of anaphase as the chromosomes are migrating towards both ends. And here's pretty much the late anaphase getting in towards telophase. And don't forget, during telophase, our nuclear membrane will start to reform. So here's our nuclear membrane going around. There's our nuclear membrane. And now the cell, after telophase, the cell is going to start to pinch in. And this pinching in of the cell membrane is called cytokinesis. Division of the cytoplasm, protoplasm, basically all the organelles. And the cytoplasm is going to divide usually down the middle, and it's usually aided by spindle fibers. There's these spindle fibers that actually close up and cinch up around the cell membrane and help close it up and divide it. And now you see that we have two cells, and look what we have. We have two genetically identical cells. Remember, the parent cell had two chromosomes in it, that blue one and the red one. And now we have two cells with that same number of chromosomes and that same exact copies of chromosomes. Every gene on those chromosomes is genetically replicated. And so we call these cells diploid cells. Both of these are 2N cells. They are diploid, and they are genetically identical. So they are diploid. And we have two of them. And that's because the DNA was duplicated or replicated. And remember, if you replicate something, you make a copy of it, you have double of everything. You have a copy of everything. And then if you divide it in half, like you did during cytokinesis or mitosis, really, and then cytokinesis, of course, then you go back to the original that you have. You have the same amount. Right? And that's how all of our cells in a human have 46 chromosomes, and they've all divided by mitosis, with the exception of the sex cells. But all somatic body cells have done mitosis. Okay, well, I hope this makes understanding about the chromosomes and mitosis a little bit more understandable. And don't forget, those somatic cells are diploid. Okay. All right, and I hope you learned something, and I will see you in class. All right.